All right, so today is the second to last lecture. Today we're going to be doing chapter 19, which is population and community ecology, and then we'll talk about chapter 20 on Wednesday. On Wednesday, that is going to be our last lecture. Reserved the last few weeks primarily for you guys to wrap up assignments rather than be presented with new material. So I will have some office hours available next week where I will be present in a WebEx meeting, but there just won't be any lecture slides presented. So I am available if you guys need help on the research paper or any of the material, but no new material will be presented after this week. So let's go ahead and just define what a population and a community is first. So a population is a group of organisms of the same species that live in the same area, and so they have a potential for interbreeding because they are in that same area. So that's what we're talking about when we're talking about a population of organisms, that same species in that same area. But then when we're talking about a community, we're still talking about a given area, but we're talking about all of the populations in that area. So the plant species, the animal species, the microbe species, all of those are populations in a given area and make up that community. They're all of the biotic or living things in that community. So when it comes to populations, there are a couple of ways that they can be studied. So one of the main things to study about a population is to study how many there are in that population. So the population size. What's the total number of individuals in that population? It's important to know this because the population size affects the amount of genetic variation present in a population. And if the amount of genetic variation is limited, that can limit the potential for adaptation. So if you have a really small population size, it's not great for your genetics. So scientists really like to characterize these population sizes so they can determine, if a population is big enough to, carry on for really, really long periods of time genetically. But it's also important to understand the population density as well. So this is the number of individuals within a certain amount of area. So, we might have a population that's in one general area, but if most are congregated in part of an area as opposed to the rest of it, that can affect things as well. So it affects the types of interactions that happen within that population, specifically competition, right? If most of our population is congregated into this one part of the habitat, there's going to be a lot more competition for food and potentially a lot more competition for mates that's happening in this more dense area. So we want to understand how many individuals are there and how they're kind of spread out within their habitat. So when it comes to determining the population size, probably the most obvious way to do this would just be to go out and count them all, right? We'll just go out, we'll count everybody in the population, and then we'll know what our population size is. But this is not a feasible thing to do. Just logistically speaking, you're never going to see every single organism, and it's just going to take too much time. It's just not worth it. So what scientists do instead is they take a sample, a representative portion of the habitat, and then they can kind of estimate things or make inferences about a larger population. Right? I'm going to look at this small section of the habitat or multiple small sections of the habitat, and then I'm going to just kind of infer things about the larger population. This is why we have statistics, right? Statistics allows us to take a sample of the population and make inferences about the larger population. There's two main ways that scientists do this. There's lots of ways they do, but we're just going to talk about two of them. So one is using a quadrat. So this is for organisms that are stationary, like plants, or maybe they're very, very slow. Maybe we're studying like a snail or something. So what you do with that is you take this plastic square or like a PVC pipe square, or maybe it's like a metal or something like that, and you put it down in the habitat and you count, right? So we are kind of getting at that, just counting the number of organisms, but we're looking at this very small space. So we're going to take our square, we're going to put it down, we're going to count all the organisms in that square, then we're going to pick our square back up and we're going to put it at other random locations in the habitat many times and then estimate the population size and density from that. So this is a picture of one of those here. We were interested in our dandelion population or maybe our clover population. There's some clovers in there as well. We could just count them all that are in this square, pick up our square, put it in a new place, count them again. We're getting an estimated number and density in this square. So then we can just make inferences about the larger habitat that way. But this only works if organisms aren't really moving that much, but a lot of organisms do. So if you are a mobile organism, generally what scientists are going to do is they're going to do what's called a mark recapture method. So you capture some of your population, you mark them somehow. This might be a little tag. You can see this mouse has a little tag on his ear. You might mark them with maybe a little bit of some sort of animal-friendly paint. Whatever it is that you're doing, you are indicating that you have caught this animal before. Then you are going to release them back out into the environment. And then you come back, you take a new sample, and you kind of see how many of your marked animals are in that sample. Okay, so this is often used for things like mice or birds, but it can also be used for whales. So certain species of whales have individual flukes. So this is called a fluke ID. You look at the fluke of the whale, and that pattern and the way that the fluke is designed on the outside tells you who that individual is. So scientists can look at a whale in maybe Hawaii, and then they can see that whale maybe again in the feeding grounds in Alaska and look at estimates for how large a population is based on how many individuals they're seeing over and over again. So there's variations on this, but basically we capture organisms or we mark them in some way, and then we come back later and see how many of the individuals that we see in our new capture we've seen before. So there's a little math involved in figuring out the total population when we're doing mark recapture. So if we think about just sort of this as a proportion, right, the total number that we mark in the first catch is a certain number out of the total population. And then when we come back and we do our second catch, what we're working off here is the idea that the proportion that are marked in the second catch are the same proportion of the total number that have been marked. So the number marked in the first catch divided by the total population, this proportion here is going to be equal to the total number marked in the second catch, or divided by the total number in the second catch, right? How many of this second catch are marked? So if we rearrange this, we get this equation here, because what we're looking for is our total population size. So if we just rearrange our equation, our total population, or N, big N here representing our total population, is going to be the total number marked in the first catch times the total number in the second catch divided by the number marked in the second catch. And we're just taking that first equation and we just rearranged it. So if we think about an example, so let's say we went out and we captured 80 mice out of a population. We marked them somehow with a tag and then we released them. We came back and then we captured 100 mice. Of these 100 mice, 20 of those are marked. Okay, so we just plug all of this into our equation. So we're going to do 80 mice that are captured in the first catch times 100, the total number in the second catch, and then divided by 20, the total number that are marked in the second catch. And then we get a total of 400. So our estimated population size is 400, right? Because again, this is just a proportion. We're saying that 80 out of 400 is equal to 20 out of 100, right? The total number of that are marked in the first catch are that proportion out of the population is equal to what we're seeing in the proportion in our second catch. So in addition to studying the number, right, with our quadrat or with our mark recapture, we also want to study that distribution, that density. So we can look at this with a specific type of species distribution pattern. And there are three primary species distribution patterns that are seen in nature. So the first one is just random, right? There's no real rhyme or reason to why these species are distributed the way that they are. This is primarily going to be like plants that have wind dispersed seeds, right? A dandelion. There's no real rhyme or reason to where they land. They just kind of happened to land there. It's completely random. Okay, but one of the more common ones we see is clumped. Clumped is probably the most common type of species distribution pattern that we see, where we have a group of organisms that have clumped together and are staying clumped together. So we've got this herd of elephants here Anytime there's a herd of animals that are traveling together, that is a clumped distribution. They might be congregating around resources that they need, or maybe they're congregating for some sort of mating activity, right? Clumped is a very common type of distribution pattern that we see. But it's also possible to see a uniform distribution where individuals in the population are equally spaced out. So this is different from random, because random, there was no real rhyme or reason to where the organisms were. But with a uniform distribution, they are equally spaced out. So here in that picture, you see penguins that are, they have their specific territories that they are, rearing their eggs on. So they are evenly distributed. So this is typically going to be some sort of territory situation. Sometimes there are plants that secrete out specific chemicals that prevent other plants from growing nearby. So that might be another example, but something is preventing them from being closer to one another. So here's just some pictures here of different types of distribution patterns. So let's just talk about these really quickly. So up here in the corner in the dandelions, it's very similar to what we saw before. This is going to be a random distribution, right? We've got some little clumps over here, some are kind of by themselves, but if you were going to go out and describe how these dandelions were laid out in this field, you're just going to say it's kind of random, right? You just kind of like throw some seeds out and you see what you get. Here we've got a herd, so this is going to be a clumped distribution, right? Anytime you have a herd of animals, that's going to be a clumped distribution. Down here we have a scientist studying some albatross, so this is going to be, you see how they're all just kind of all equally spaced out, so very similar to the penguins that we saw before. These are a uniform distribution. Here in this photo of this suburban neighborhood, this is also a uniform distribution, right? Each individual house has its own set of land that's equally spaced from one to the next. So this is a uniform distribution. But if we kind of back up and look at a bigger picture of human distribution, which is what you see here, this is the east coast of the United States, you can very much see that human population is clumped, right? We clump around these urban areas, and then it's kind of more distributed outside the urban areas. So human distribution very much clumped, tend to clump in urban areas. These urban areas are often congregated. Maybe like Denver is right at the entrance to the Rocky Mountains. New York is a really active port, just like Los Angeles and San Francisco. Chicago congregated right around that lake. So clumped distribution for human populations. So when scientists are studying populations, studying population size and density is super important, but this is only looking at a specific point in time. So in order to look at a longer time scale, they have to use something called demography. So demography is just this quantitative or statistical study of populations, looking at how they change over time. So we're looking at the births that are introducing new organisms into the population, immigration, bringing new animals into the population, and then also looking at the death rate and then the immigration of individuals leaving. So when we talked about the types of microevolution with gene flow, we talked about immigration. So immigration with an I is going to be individuals moving into a population. Immigration with an E is individuals exiting a population. So we have to look at that migration, and we have to look at the birth rate and the death rate. So all of those factors that are affecting how a population is growing. And with this, they use two primary methods. So there are things called life tables, and then there's also something called a survivorship curve. So a life table, this is just a table of information that includes mortality rate and life expectancy. So it's often broken up into the age range or the cohort range of the organisms. So here we're looking at ground squirrels. So you can see this far left column is the age group. So we have 0 to 1, 1 to 2, all the way up to 9 to 10. So this is going to be how scientists divide up the information that they're seeing. So life tables can look a little bit different depending on what type of information is being included, but in general, we're going to have how many are alive in the population within that age range, the proportion that are alive at the start of the year. So of our individuals that are 0 to 1 years old, none of them have died yet. So there is 100% of those still alive. So that's why our proportion alive at the start of the year is 1. But if you go to the next line here, we started with 337. Now there's only 252 in this next age range. So the proportion alive at the start of the year has decreased. Okay. So we also have the number of deaths during the year and the corresponding death rate, all of this to kind of get the average life expectancy of these organisms. So as they proceed through this life table, as our age increases, our death rate is increasing, our proportion alive is decreasing, our average life expectancy is decreasing for that organism. We're not expecting them to live much longer in this age range as they get older and older. The survivorship curves is taking a lot of information from these life tables, but it looks at the number of individuals that survive at each age, and we look at that over time. So we can compare different populations, we can compare different species, and these survivorship curves kind of give us some information about the life histories of an organism as well. Maybe, do they provide parental care to their offspring? Do they not? Kind of those types of information we can get from a survivorship curve as well. There are 3 types of survivorship curves, type 1, 2, and 3. So in a type 1 curve, we have a very low mortality in the early years and middle years. So this is going to be most mammals, and humans are included in this as well. We have very low mortality in the early and middle years of life, and then as we get older, mortality starts to increase. Okay. So the reason that we typically have this low mortality is because we are in general producing not very many offspring every year. So the offspring that we do produce, we're caring for a lot and making sure they are safe and trying to make sure they are surviving and getting to those reproductive years. So this very low mortality in early and middle years. Type 2 has a constant mortality, so you're just as likely to die as a young individual as you are an older individual, so this constant mortality throughout life. And then type 3 is kind of opposite of the type 1. We have very, very high mortality in the early stages, but once you make it past a certain point, you've got a good chance of living for a lot longer. So this is typically organisms that produce a large number of offspring. So like plants or a lot of marine invertebrates do this, where they just kind of produce, hundreds, thousands, maybe even up to hundreds of thousands of offspring, but they're not putting any investment into those offspring. They're just kind of throwing them out into the world, crossing their fingers that some of them make it. So not a lot of them are going to. So that's why there's a large proportion of them that do not survive to the next age range. When we plot these on a graph, this is typically what they look like. So we've got our type 1 up here in the blue. And if you look at these axes, this is just time. So this is over time here. And on the y-axis, we have our number of individuals surviving. So our type 1 curve, we've got a high number of individuals surviving. As we go through time, it's still a high number of individuals surviving. But as time goes on and we hit these older age ranges, we start to have a really high rate of mortality. Type 2, that constant mortality is just going to be a straight line over time. But then in type 3, where we start with a large number of offspring, but not a lot of them survive, we have really high mortality at an early age. We're going to see a steep drop off here. But then once you make it down here, mortality is pretty low, right? If you can establish yourself as a tree and grow into a tree, you're probably going to be a tree for a long time, but it's just not very likely that you're going to go from a seed to a tree. Okay, so these are the 3 types of survivorship curves here. So when we track these population sizes over time, when we're looking at the growth rate and the death rate, we can look at population growth patterns, how these populations are growing over time. There are 2 main models that we use to look at population growth. There's an exponential growth model, and then there's a logistic growth model. So as we talk about these 2 models, there is going to be a little bit of math. It's not super important that we're able to use the math, but it's important that we understand the math to understand how these populations are growing. But it is also important to understand that these models don't really represent the natural populations. Very few natural populations follow these models exactly, but it's a really good point of comparison for us, right? Because we can kind of compare these natural populations to our models and kind of see what factors are influencing population growth and how it might grow in the future. So our exponential growth model, this is a population that is growing without limits. There's no cap for how many individuals can be added to this population. And when we track this, it's going to be a J-shaped growth curve. So this is our growth curve over here in the figure. We've got our population size over time. And you can see it's kind of a J shape, right? So we start out pretty small, but we're adding more and more individuals. And as we grow, the amount of time it takes us to add more individuals shortens because we already have more individuals, right? So there's been lots of talk of exponential growth in regards to the coronavirus, and we want to make sure that we are, flattening this curve. We want this exponential growth to stop. So that's what this has been compared to recently. The population growth model for this is this formula here, this RN formula. It's just the rate of growth, how quickly they're growing times the number of individuals. It's a very simple formula. We're just taking our rate, which our rate is going to be some combination of birth rate and death rate, right? And we have to factor in both of those things to get a growth rate. We're just going to multiply that by how many individuals, and then we can model our population growth. If we think about this in terms of just R with our rate of growth, if this is a positive rate of growth, our population size is going to be increasing, right? Our growth is going to be positive because our R value is positive. N is always going to be positive because it's our number of individuals. We can't have a negative number of individuals. If our growth rate is negative, our population size is decreasing, right? And that negative number is going to give us a negative population growth. So our population size would be decreasing. But if our R value is 0, our population is staying the same. There is 0 population growth. Okay, so that R is really important for us to understand what's happening to the population. We want to understand this rate of growth so we can see if our population is increasing or decreasing, or maybe it's staying the same. With our logistic growth, it becomes a little bit more complicated because there is a limit to the population size, and this limitation becomes more intense as the population size increases. So instead of a J-shaped growth curve, we're going to get more of an S shape. We can see this kind of S shape in our figure here, and it's limited by what we call the carrying capacity. So carrying capacity is often represented by the letter K, and this is just referring to the maximum population size that the environment can hold. So as we increase towards this maximum population size, this carrying capacity, our population growth is going to slow down, and then we're eventually going to reach our carrying capacity. And we're eventually going to hit that maximum value of organisms that an environment can hold. So our equation looks a little bit more complicated. We still have our R and our N, right? That's exactly the same as our exponential growth, but we've added this other bracketed amount here. And this is just allowing us to take into account that carrying capacity. So this is how we do that, right? We have our rate, we have our population size, but then we have this that's allowing us to include our carrying capacity. So what I want us to think about with this, again, it's not important necessarily for us to be able to use these equations, but it's important for us to understand what happens to the population growth if certain aspects of this equation are altered, right? So we're talking about our carrying capacity here and our N, which is the number of individuals we have in the population right now. So if N is close to 0, right? If our population is really, really small, if our population size is close to 0, then our bracketed value is going to be close to 1, right? If N is close to 0, then K is basically not changed that much, and then K divided by K is going to be really close to 1, right? Because K isn't really changed that much because our N value is so small. So if this bracketed value is close to 1, then our population growth is going to be just like an exponential growth, right? This bracketed value is 1, so it's really just this R times this N. which is exactly what our population growth model is for exponential. So you can see down here, when our population growth is really, when our population is really small, our growth kind of resembles this exponential curve, okay? As our n gets bigger, if n is very close to k, if it's almost the same, if we're really close to our carrying capacity, what's on the top of our fraction here is going to be really close to 0, right? If n is very close to k, that top value is going to be close to 0, and then 0 divided by k is going to be it's going to be 0, right? So if this bracketed value is 0, this whole population growth is 0, right? So as we are really close to carrying capacity, our population growth tends towards 0 growth, right? We are close to that carrying capacity. We can't hold very many more organisms in the environment, so our population growth is going to be basically 0, right? So it's going to be important for us to know how to manipulate these equations depending on how many individuals are in our population. So this is a much more realistic model than the exponential growth, because in general, there's going to be some sort of cap on the population. But typically in the natural populations, they don't just gradually increase towards the carrying capacity. Typically what happens is they go past the carrying capacity, they overshoot that carrying capacity, and then once we are above the carrying capacity, that environment cannot sustain that number of organisms, so our death rate is going to increase past our death rate, and our population is going to come back down. Our R value, our rate of growth, is going to be negative. Our population will decline back down towards the carrying capacity, and then we kind of undershoot the carrying capacity, which allows us to grow more, and then we kind of overshoot and undershoot, and it just kind of oscillates around that carrying capacity until it levels off. And it's also too important to think about competition when it comes to carrying capacity, because it's the environment that can hold a maximum number of individuals. But as this number of individuals in our population grows towards this carrying capacity, competition between members of a population becomes much more important, right? Intraspecific competition. Competition within your species. So intraspecific within one species, that type of competition. Because you're using the same resources as your population, right? Other individuals in your population. You're eating the same thing, you're living in the same place, you're competing for the same mates. So as there are more individuals, that competition becomes more intense. And it's also important for us to know that K, the carrying capacity, is not constant. Maybe the environment changes and it can now hold more organisms, or it changes and it can hold less organisms. That carrying capacity can fluctuate. So populations that are impacted by this carrying capacity and are aiming towards this carrying capacity might not be aiming for the same population size all the time, because K might change. Maybe it changes even within a year, right? Maybe an environment can hold a lot of organisms in the summer, but in the winter, it can't hold that many. So even within 1 year, that carrying capacity can change. So here we just have some examples here, kind of breaking down what these growth models look like. So in both of them, we see this lag phase. You can see that in both of those figures there. And this is just because it takes a while for us to really get the ball rolling here, right? If you think about like a snowball that you start to roll up, it's really small at the beginning, but as you add more and more snow to it, it gets really big really quickly. So once it's getting really big really quickly, that's the exponential growth phase there. So we have that over here in our exponential growth, but then we also have that over here in our logistical growth. But as we keep going up, when we have a carrying capacity to think about, we're going to start to slow down our deceleration phase. Then this is what we're doing. We're overshooting the carrying capacity, then we're undershooting the carrying capacity, then overshooting and undershooting. So we're just kind of in this stable equilibrium, or this oscillation phase, where we're going up and down around the carrying capacity. So understanding population growth and how it's regulated is important for us to think about, because scientists are very interested in preventing the extinction of certain populations. They might also be interested in preventing overpopulation of some groups. So we need to understand how our population is growing with those models, but then we also need to understand how our population is regulated. So what's impacting this type of growth? So there are two general types of factors that we need to think about when it comes to population regulation. And there are factors that are density dependent, so it depends on the density of the population. And there are some that are density independent. Density dependent factors are going to be factors where the mortality increases or birth rate decreases as the population density increases, right? So as our population increases, these growth regulation factors are going to become more intense. As our population grows, our death rate is going to increase or our birth rate is going to decrease. So this could be predation, right? As you are becoming a larger population, it's going to be much easier for predators to eat you because there's more of you, right? Easier to catch if there's more of you. Competition is going to be important to consider. We talked about intra-specific competition as our population size increases, specifically referring to this density. As the number of individuals in a certain area increases, the competition for resources is more intense. If the competition for resources is more intense, you might see a higher mortality rate, a higher death rate, and you might also see a lower birth rate. Okay? Parasites could also be a type of density dependent factor, just something that is going to become more intense as the population density increases. So you can see here, we've got some examples. So we've got our bird population here. We've got the number of breeding pairs and then the average clutch size. So the number of eggs, the average number of eggs that's being produced when there's a certain number of individual birds in that population. When we have a low number of breeding pairs, we have a lot of eggs produced in a clutch. But as this population increases, we see a decrease in the number of eggs that are produced. So this is a density dependent factor that's at play here. We also have another example. We've got some beetles, we've got our density, and then this is just reflecting the percent that are surviving. So as we increase in density, the number of individuals that can survive is decreasing. So a decreasing birth rate with increasing density and an increasing death rate with increasing density. Here's another visualization of this density dependent factor. So let's say we've got this bird species that lives in hollowed out tree trunks. So when we have this number of tree trunks in a population, if the number of birds is less than the number of trees, each bird has a little house, right? There's not a lot of competition. All the birds are nice and safe when the population is small. But as that population increases, all of our tree trunks are going to be filled. So we've got birds that are out here without a place to live in those tree trunks. So we've got a high competition for those tree trunks. We would also probably see an increase in predation. So that when that population is large, this is a density dependent situation because it depends on the number of birds that are in that area. Density independent factors, however, are when the mortality rate does not depend on the density. So this is going to be something that is typically not related to the biology at all, right? It's some sort of weather event, natural disaster event. So here, this is looking at an insect population, number of aphids. So we've just got the months of the year plotted here. We see an increase during the summer and then a sharp decline in the middle of the summer going into the fall. So this is just the cyclical nature of the seasons. So they have a high population in the summer and a low population in the winter. So this is completely independent of their density. It doesn't matter how many individuals are in this population in this area at this time of year. No matter what the size of the population is, they're going to decrease because the weather is changing. Okay, so completely independent of the density here. Natural disasters would be another example. If you have a population and a tornado comes through and destroys all of the habitat, that's completely unrelated to the density, but can really impact that population growth and can cause a decrease in that population density. And then some population regulation can result in these really interesting cycles. So this is the classic example of cyclical population growth with some predators and some prey. So this is specifically referring to the Arctic hare. You can see in this picture here, that's represented in the red on the graph, and then the lynx over here, which is represented in the light blue on the graph. So this is looking at the number of animals over time. So you can see back here in the mid 1800s, we had a little bit of an increase in the lynx population. So that the amount of prey, the hare in the population was so small that the lynx population couldn't be sustained. So that population then decreased. So once that population of lynx decreased, the hare were able to rebound and increase in population because they didn't have as many predators coming after them. So we see this increase in the hare population. Once we have this increase in the hare population, we're able to get more predators, right? So the predator population then comes up. But then once the predator population comes up, the hare population decreases. When the hare population decreases, the lynx population decreases. So we have this cycle that's happening. You can see it's really well illustrated in these last few peaks, where we have an increase in the hare population, which causes an increase in our lynx population. But once that lynx population increases, our hare population decreases, causing the lynx population to decrease. Hare population increases, lynx population increases, hare decreases, lynx decreases, right? This population regulation between two populations because of these predator-prey dynamics that are happening. And then one other thing that I want to touch on with regards to population growth, scientists often classify different types of species into how they grow, and the way that their population grows often comes hand in hand with some other reproductive strategies of that population. So there are two types of species that scientists use to classify. There's something called a K-selected species, and there's something called an R-selected species. So these Ks and these Rs are coming back from our population models with our rate of growth and our carrying capacity. And then that table there shows the types of strategies that these organisms have. So in general, our R-selected species have a short life. They're growing really fast. They have lots of really small offspring with not a lot of parental care. They are generally going to be, if you go down here to the bottom, they're generally going to be the prey species, and they're generally going to be occupying a low trophic level, which means a low rank on the food chain. Okay, so those are generally going to be our R-selected species. But our K-selected species are going to kind of be the opposite end of that spectrum. They're going to have a long life. They mature much more slowly. They might only have a few offspring, but they put a lot of investment into those offspring. There's a lot of parental care there. And if you go down here, these are generally going to be our predators. They're not going to be the prey species, and they're generally occupying that high trophic level. So thinking about the life history strategies of these populations, we can group them into either being R-selected or K-selected. So R-selected would be maybe a lot of plant species, some microbial species, some marine invertebrate species. But a K-selected species are going to be, like the classic examples are elephants, whales, people are K-selected. But if you really think about all the species that are out there, this is going to exist on a spectrum, on a continuum. So at the R end of this spectrum, here we've got one of our bivalves from our mollusks that's producing 500 million offspring a year, right? They're not putting any investment into these offspring. They're just casting them into the environment. But then down here on the case end side, we've got our gorilla, which are producing one offspring every 5 years. Okay, so most species are going to be somewhere in the middle of this spectrum, but in general, we can classify things as either R-selected or K-selected, depending on those life history strategies that they have. When we think about the human population, we can incorporate a lot of the same ideas that we've talked about already, and we can apply them to the human population. The interesting thing about humans is that we've altered the environment to alter our carrying capacity, and we've done this many times. Okay, so the Earth was, unaltered, can hold X number of humans. But then humans came along and invented agriculture. So that allowed us to increase our carrying capacity because we're increasing the amount of food that's available to us. Then we keep going, agriculture is helping, but and this happened, I can't remember when this happened, but we had agriculture, but then the invention of things like herbicides and pesticides allowed us to increase the productivity of our agriculture, which allowed us to increase our carrying capacity even more. The Industrial Revolution allowed us to increase our carrying capacity even more. So as we get towards our carrying capacity, humans tend to move the carrying capacity in some way, right? We tend to alter our environment to alter our carrying capacity. But right now, at this point in time, the human population is growing exponentially. So you can see here, this is just over time. You can see that it was a very slow growth, this lag phase, but then we started to see this exponential growth. And this is just kind of zoomed in. So we didn't reach 1 billion humans until 1804. But then in 1930, we hit 2 billion. And then in the 1900s, we've just been increasing billions and billions. And then we hit 7 billion in 2011. So as we grow, it takes less and less time for us to add another billion humans to the planet, right? So the first billion years took a really long time. The second billion years only took about 130 years. The third billion took about 30 years. 15 years later, we hit 4 billion. 12 years later, we hit 5. 12 years later, we hit 6. 13 years later, we hit 7 billion, right? And it's estimated that we'll hit 8 billion and 9 billion as well. So as we are increasing in this population, we're adding more and more humans, and it takes less and less time for us to add another billion humans to the planet. One interesting thing that we do with the human population is we build these age structure diagrams, and this is generally done for various countries. That's typically on the scale that you see it, is an age structure diagram of the population of one country. And this allows us to better predict future growth. Basically, what happens is we take different cohorts, different age ranges, and we plot the number of individuals in those groups in these little pyramids. So so in this example right here, the stage 1 age structure diagram, we have lots of individuals in these low age ranges, but as we increase in age, it thins out pretty heavily, right? We only have a few individuals in these higher age ranges. These are typically going to be seen, these stage 1 age structure diagrams are typically going to be seen in developing countries. That's typically what you see, where you have a lot of young individuals in the population, but it thins out pretty quickly. There's also this stage 2 slow growth, where we have more individuals in these lower age groups, and as we increase in age, they do decrease, but it's just not quite as steep of a decrease. So individuals are able to live longer in this slow growth stage. Stage 3, this is stable, where you kind of see they're almost flat on the sides here, indicating that there's approximately the same number of younger individuals as there are a little bit older individuals. We don't really start to come in and decrease the number of individuals until we're older. So this is something that you see in a population that's not growing too fast. So Italy is a good example. They're not growing at all. I don't think Japan's population is growing. The United States is growing very, very slowly. So this is going to be more developed countries that see a stable age structure diagram. And then this is more of a theorized stage, this stage 4, where we might start to see fewer individuals in the lower age range compared to a higher age range. And this is primarily because individuals might not be having as many children. So if individuals aren't replacing themselves with offspring, we will have a smaller number of individuals in those lower age groups, and we'll have a smaller, like a decreasing population, basically. So with these lines that are coming in, you would see a decreasing population size here. And this is kind of how it plays out when we look at all the countries compared to one another. In general, the highest population growth, those stage 1, stage 2 types of age structure diagrams, are going to be in developing countries. So we can see the highest population growth, this 2%, 3% growth, are primarily going to be in developing countries. A lot of our 1% population growth is primarily in developing countries. But then our much lower population growth, so in the United States and in Europe, and in some parts of Asia and South America and Australia, we have a very low population growth. And then there are some countries that even have a negative population growth. So kind of that stage 4 age structure diagram, where not all of the individuals in that population are having children, therefore the population size is decreasing. All right, so let's move from populations into communities. So our populations, well, we've talked about a lot of dynamics within those populations. They're not isolated. They're interacting with lots of other populations to form this community. And when we talk about the interactions between one population and another population, these two different species that are interacting, these are called interspecific interactions. So we mentioned intra-specific competition. Intra is going to be within one species. Interspecific is between species, right? An interstate goes between species. That highway that, or not species, interstate connects states, right? Interstate highway is going to be between states. So interspecific interactions are between species. And we can classify these types of interactions by how they impact the populations that are involved. And these interspecific interactions are also called symbiotic relationships. So symbiotic, living together, these relationships that happen when they are living in the same place. So this is a summary of the interspecific interactions we're going to talk about and the effects that they have. We're going to go through each of these individually, but this is going to be a nice summary. When you're studying, you can kind of quiz yourself on the different types of effects that these interactions have. So predation, this type of interspecific interaction, is a plus-minus interaction, meaning that it's going to benefit one of the individuals involved, but it's going to be harmful to the other individual involved. Okay, so one species, the predator, is going to kill and eat another, which is the prey. So we've got a bird eating a caterpillar here, a tiger that's got a piece of meat. We've got our predators, we've got our prey. Clearly, one is benefiting and one is being harmed in the process. With our predator interactions, we see a lot of really interesting adaptations that predators have. So most predators are going to have senses that are highly acute or very fine-tuned. They might have other things that benefit them as well, claws or teeth, maybe fangs or stingers, poison, a lot of these things that help them catch and subdue their prey. So they have these adaptations that they've evolved to be predators. On the other side of that equation, we also have prey that have evolved defenses against our predators. So obviously, natural selection is going to favor a way for you to defend yourself against predators. That is going to, if you have a defense, you are going to be able to survive and reproduce more than somebody that doesn't have a defense. So natural selection is going to favor those individuals. That might be hiding. So we've got an example here of a frog that's camouflaged. You might just be quick and be able to actively escape. You might defend yourself like this porcupine here. There might be some sort of alarm call. If I can communicate to members of my species, my population, that there's a predator nearby, we can escape more easily because we know it's coming. Mobbing might be another example. So where we are actively retaliating against our predators. So those are all types of defenses against predators that we see in nature. Some animals also have some active defenses here, some mechanical or chemical defenses against predators. So monarch butterflies taste really bad, so birds tend to avoid them. Skunks can spray. We've got a poison dart frog here. They have these active defenses against predators. And often these chemical defenses or mechanical defenses come with some sort of warning coloration, right? I am brightly colored. Do not eat me. I am brightly colored. Do not eat me. I am poisonous. Don't eat me. Skunks have this very distinct coloration pattern that makes them very easily recognizable. So predators, if they've encountered a skunk once and been sprayed, and then they see a skunk again, they might avoid it because they recognize that really distinct coloration. So sometimes, because of this type of warning coloration that's very effective in communicating to predators, a lot of times multiple species that all have defenses might have a similar coloration. So all of these butterflies kind of have a similar coloration and it's communicating the same message to predators. We all have this coloration. We all taste bad. Don't eat us. That's why we look this way. Other individuals may adapt this warning coloration even if they don't have the same defenses as an individual that does have those defenses, as a way to kind of trick predators. So here we've got a type of fly, a hoverfly, I think it's called, that looks very much like an insect that could sting you. This is actually a harmless insect right here that's adapted this warning coloration to try and fool predators. So like we talked about that monarch butterfly also, there's another type of butterfly that looks very, very similar to the monarch butterfly, but doesn't actually taste bad. It just makes birds think it tastes bad so it can avoid getting eaten. Either of these strategies are both examples of what's called mimicry, where you are mimicking another species in order to communicate to the predator that you should not be eaten. Whether that's all of you have defenses and look the same, or you might not have any defenses and you just look like somebody that does. Those are both examples of mimicry. Herbivory is very similar to predation, right? It's still this plus minus interaction, but rather than one species killing and eating what is called a prey animal, this is where one species, an herbivore, is eating a plant, right? It's still killing and eating the organism, it's just a plant rather than an animal. So very, very similar to predation. And just like our prey had defenses against predators, plants can also have defenses against herbivores. So this could be a chemical toxin, but it might also be just a mechanical defense, like thorns or spines. That can be a defense against predators. And then we also have things like poison ivy that are defenses against predators as well. Parasitism is another plus minus interaction. So similar to predation and herbivory in the sense that it's plus minus, but in this type of interaction, we have one organism that is benefiting and the other, which is called the host, is harmed, but our host isn't immediately killed. Our parasite, which is what is benefiting in this interaction, needs to use the nutrients from the host. So it doesn't want to kill the host because it still needs to suck down all those nutrients. So it's going to keep the host alive at least for a while until it can reproduce and move on to another host. So here we have a moth that's been infected with this type of fungi. All of these spikes sticking out of it are a parasitic fungi that have inhabited this host. And then here, this is an example of brood parasitism where one type of bird will lay its eggs in the nest of another type of bird, and then that bird is tricked into raising that young. So a little bit different type of parasitism, but this individual is still negatively impacted because it's wasting all of this energy raising young that is of a completely different species. The point of it spending so much time to raise its young is to put its genes into the next generation. It's not doing that right now. This is just wasted energy on the part of this individual. So this species is benefiting here because this chick is getting reared by someone other than the parent. The parent can go off and make more chicks because it's not it's wasting its time, not wasting, but spending its time raising this individual, and then this one is negatively impacted. All right, moving into competition. So we're no longer in a plus minus interaction. This is a minus minus interaction. Anytime we have competition, that is going to negatively impact both parties involved. So this is where two or more species are trying to obtain the same limited resource. No matter how you look at competition, it's never going to really benefit anybody. It's just going to cause both parties to expend energy to obtain this resource. Obtaining the resource is a positive result, but competing for the resource itself is a negative impact. And the population growth of species, when we're thinking about competition, can be limited by how many individuals are in its own species and how many individuals are in that competing species. So we have to think about both of those densities when we think about competition. An interesting thing that happens with competition is something that we call the competitive exclusion principle. This is basically saying that when we have two species that are competing for resources, if they are occupying the same niche in a habitat, so this niche is a unique set of resources, if they are using this exact same set of resources, eventually one is going to outcompete the other. Two species cannot, for an extended period of time, occupy this exact same niche in a habitat. They can't use the exact same set of resources in a habitat. One is always going to outcompete the other. So the classic example of this, it was an experiment done in the lab with two species of Paramecium. So we have one species here that when we grow it in the lab, we see this type of population growth here. This is a second species that when we grow it alone in the lab, we see this type of population growth. So very similar population growth between these two Paramecium. And when they're being grown in the lab, they have the exact same amount of resources, right? Species number one and species number two have the same amount of space, the same amount of food when they're growing. So if you take both of those species and put them together and give them the same amount of resources, this is what's going to happen. Okay, one of our species is able to outcompete the other species. They eat the same thing, they use the same resources, but one of these species is going to be able to grow and the other is not. The competitive exclusion principle states that two species cannot occupy this same unique set of resources in a habitat. One is going to outcompete the other. So there are two outcomes of competitive exclusion. One is that the species that is outcompeted goes extinct. It can't survive in this habitat anymore because it doesn't have access to this specific set of resources. So one species is going to go extinct. The other option is that one species may be outcompeted, but it's going to evolve to use maybe a slightly different set of resources than it was using before. Okay, so this is often referred to as resource partitioning. And a good example of this is with the species of warblers that we see. So we've got 5 species of warbler. They all occupy similar niches, but they have kind of partitioned the resources so that one type of warbler is using one type of resource, one aspect of this resource, and the others are using other aspects of this resource. So maybe all of these warblers eat the same thing and live in the same tree, but rather than competing for the whole tree, they've decided to partition this tree into different areas. So our yellow-rumped warbler is going to live at the bottom of the tree. Our bay-breasted warrior is going to live in this middle part of the tree. Our Cape May warbler is going to live at the top of the tree. Okay, they've partitioned these resources in order to coexist in a habitat. So none of these warblers are going extinct, but they have kind of adjusted the resources that they're using in order to coexist in a habitat with other species. Commensalism is another type of interspecific interaction. This is where one organism is benefiting and the other one is not affected really at all. So it's not a negative interaction, they're not harmed, but it's not really a positive interaction. They're not getting anything out of it. So they're just kind of unaffected. So a great example of this is birds nesting in trees. Okay? Birds are obviously benefiting, the tree's not really affected. So that's a commensalistic relationship. This down here, this little fish that has attached itself to the shark is a remora fish. So these fish like suck onto the sides of sharks and just hitch a ride. So that shark's not really affected. It's not harmed, but it's not really benefiting. But that remora can just kind of latch on and get a free ride. So it's obviously benefited because it doesn't have to expend energy moving from one location to another. That shark is able to move at a much quicker pace. It's much larger. It can cover larger distances. So that remora fish is just going to hitch a ride and move on to a new habitat without expending any energy. And then we also have a mutualistic relationship. We talked about mutualistic relationships when we talked about fungi. This is where both parties are benefiting. So this was our fungi example here, mycorrhizae, where we have These white parts are the little threads of our fungi that are interacting with plant roots. The plant roots are providing food for the fungi. The fungi are providing nutrients to the plant roots. So both are benefiting here. It is an equally beneficial relationship here. Bees and flowers is another example of a mutualistic relationship. Bees are getting the nectar they need out of the flowers. The flowers are getting pollinated. Okay, so a mutualistic relationship. Both parties are benefiting. So the last set of things, or maybe the second to last set of things that we want to talk about with communities are just characteristics of communities. The big one here is going to be biodiversity. That's a big aspect of communities that scientists like to study. They like to see how many species and what types of species are in an area. So the diversity of species in an area is just called biodiversity. And then I just threw this map up here. It's just a really great biodiversity map of the United States when it comes to mammal species. You can see that there are certain places in the United States that have a really high biodiversity of mammals and other places in the United States that maybe have a little less biodiversity of mammals. We are really lucky here in Colorado that we have a pretty high biodiversity of mammals. So there are three aspects of biodiversity that we need to think about. One is the species richness. Species richness is just the number of species. So how many species are in a given area. But in addition to the richness, we also want to think about abundance. So this is the number of individuals in a given area, right? If you have a large number of species, do you also have a large number of individuals? Or maybe you have a small number of individuals and a large number of species, or vice versa. So we want to understand not only what types of organisms are here, but how many of each of those organisms are here. And then those kind of play into what's called species evenness. And that's just the relative abundance of the different species that you see. So evenness kind of incorporates the number of species and the number of individuals into this measurement. So it's really thinking about how evenly spread the number of individuals are within the number of species. So we've got two communities here. We've got community number 1 that has 4 different types of trees. Each tree represents 25% of the total number of individuals. Community number 2 also has 4 species, but we see down here that the percents here are a lot different. This type of tree, we only have 6% of our individuals, but this type of tree here, we've got 70% of our individuals are that type of tree. So while they have the same species richness, they have the same number of species. They each have 4 species. They have a different evenness. Community number 1 is going to be much more even because those relative abundances are more similar. So community number 1 is more even, has a greater evenness than community number 2. So if we look at pictures of communities, these are communities from the African plains. We want to think about which community is most diverse here. So in this picture, we've just got one species of animal that we can see. We've got some grasses, obviously, but really just like one species of animal here. In our bottom picture, we've got 2 species that are represented, but it's not very even, right? We've got a lot of wildebeest and only 1 lion. So we've got more species, but they're not very even. And then in this picture up here, we've got quite a few species. It's a little hard to see on my screen, but it looks like about maybe 4 species, and we've got a couple individuals in each species. So this community right here is going to be our most diverse because it has a greater species richness. It's got a greater number of species, right? And it also has a nice species evenness. It's kind of evenly dispersed. That abundance is equally dispersed amongst those species. Another characteristic of communities we want to think about in addition to biodiversity is the stability of that community. So is it able to resist change, or if it is changed, if it is disturbed, is it able to return to its original species combination? So this depends on what that community looks like, what the disturbance looks like, but the stability of that community is its ability to rebound to that original state. So if we have some sort of fire that comes through a forested area, is it able to rebound? So here we see it's starting to rebound with the addition of some new plants after that fire event. So when we have disturbances, sometimes it actually can increase our diversity. We might think that a disturbance would decrease our diversity, right? A fire is a pretty extreme example of a disturbance that is going to decrease our diversity. But if we have a really small scale disturbance, this might actually increase our diversity because it's giving other organisms the opportunity to grow. We have a really large tree, that's just one individual. If that tree falls, it might give the opportunity for multiple organisms of other species to grow and thrive. So those small scale disturbances can actually increase diversity if you think about it that way. But disturbances can also be pretty gradual. So ecological succession is the last thing we're going to talk about today. This is what happens after a very severe disturbance. So this is going to be a very slow, gradual change in species composition as a result of the disturbance that's very severe. Ecological succession, the definition of that is when we have this sequential appearance and disappearance of different species in a community over time. So things are there's a severe disturbance, some organisms are going to start to colonize that, but then new organisms are going to come in and kind of replace those colonizers. And we're going to carry on in this kind of timeline of appearance and disappearance of different species until we reach what's called a climax community, which is the final members of this community. And there are two types of this. It's called primary and secondary ecological succession. In primary succession, this is where we have brand new or brandly new exposed rock. So if we have volcanic activity that creates new rock, like lava cools and hardens to create new rocks, this happens in Hawaii. The big island of Hawaii has an active volcano, and that island gets bigger every year because of new lava that's added to that island. So that's brand new rock that's available. Glacial retreats is another one. If we have a glacier that retreats, it pretty much scrapes that rock clean. So it's newly exposed rock that's able to be colonized. So when we have these brand new bare rocks, we're going to initially get some pioneer species that come in and establish themselves. There are not a lot of species that can do this. Lichens, that symbiotic relationship we talked about with our fungi, that is going to be a pioneer species. There are some types of plants that can establish themselves in these rocky areas. The establishment of these lichens and these other plants starts to break down the rock. And once that rock starts to break down into more soil type of sediment, new plants are able to come in. So on this timeline here, we have our bare rock, we've got our lichens, we've got that rock being broken down even further to get some small annual plants, then we get some grasses. Once we have those grasses established, that soil is getting broken down. We can start to get some shrubs, some plants that like the sun, shade intolerant, right? There's not a lot of coverage here, so we need to be able to thrive in the sun. But once we have that shade established by these other species, we can start to see different types of species emerge, and we get our climax community, our final community composition that's going to be seen in this habitat. So this primary succession, we start with brand new rock and we get to our climax community. This takes a really long time, hundreds and hundreds of years to go from bare rock to trees like oak trees. It takes a really long time. In secondary succession, this is where we have a part of an ecosystem that is disturbed. We still have remnants of that community. We still have some soil that's been left intact. So this is going to be a much quicker rebound. So a wildfire, a flood, if a forest is logged, those are all examples of events that can cause secondary succession. So we have that event happen, but because we already have this soil established, we can kind of skip over a lot of the first few things that need to come in, like lichens. We don't need to break down new rock. We already have soil. So we can kind of start with some plants, some annual plants, we get some grasses, and then we see the same thing, right? We have our shrubs, and then we start to see our oak trees. So this still takes a long time, but much less time than primary succession. So primary is brand new rock, secondary succession, part of that soil is still left intact. Okay? So that is our Chapter 19 lecture. So the quiz this week is all about last week's lecture, so it's all about animals. The lab and the homework is all about animals, so make sure you get those done. The lab that you're doing today will definitely help you with the homework and with the quiz, so make sure you get that done. And then, as always, let me know if you have any questions. But Chapter 19 that we just talked about, and then Chapter 20 that we're going to talk about on Wednesday, are both going to be the information covered on the quiz next week. So I'll go ahead and wrap it up. I'll be around for a few minutes if you have any questions now, but otherwise feel free to shoot me an email.